Okay, this video is on the red blood cell, the erythrocytes. The erythrocytes basically are constructed as a biconcave disc. As you can see the dimensions, the general dimension this way, crosswise, is about 6 to 9 micromillimeters. 7.5, as you see, that's the average. But if a red blood cell is below 6 micromillimeters, then it's considered microcytic. If it's above 9, it's considered macrocytic. So about 6 to 9 on the cross section. It is a biconcave disc because that allows it to have more surface area for oxygen and CO2 to be exchanged. During its development, it loses the nucleus. When it loses the nucleus, the center can cave in, which allows it to have more surface area. And by the nucleus being lost, it gives more room for hemoglobin to be placed in the red blood cell, because it's the hemoglobin that carries the carbon dioxide and the oxygen. Here's the cell membrane, a construct of the cell membrane of the red blood cell. We will talk later about the carbohydrate portion, which is important in determining the red blood cell type, like your ABO blood type and RH. The types of proteins located in the red blood cell membrane allow it to be very flexible. The red blood cell must be very flexible to transverse through the tight angles of the blood vessels. If for some reason it loses its flexibility, it will become lodged in blood vessels, and when that occurs, blood behind that will become lodged, and all of a sudden a blood clot will develop, which could be lethal if the blood clot breaks loose. A blood clot sitting in one place is called a thrombus. If a blood clot moves, it's called an embolus. Here is the structure of hemoglobin. Hemoglobin is made up of two things, heme and globin. Heme is a tetrapyrrole ring structure, 5, with an iron prosthetic group. In order to carry oxygen, the iron must be in the +2 oxidation state. Globin is a protein in the quaternary structuring, which means it has more than one polypeptide in the chain. There are different kinds of hemoglobin, different types of globin. Adult hemoglobin, which is called HGBA, has 2 alpha polypeptides and 2 beta. You even have a fetal hemoglobin. So hemoglobin is made up of heme, which is a tetrapyrrole ring structure with an iron in the middle, hooked to globin. Each hemoglobin molecule has 4 hemes and 4 polypeptide chains. 4 hemes and 4 polypeptide chains. The average red blood cell, we calculate to have somewhere between 280 and 300 million molecules of hemoglobin in it. Okay. We go next, as I alluded to, oxygen is carried on the heme, and to carry oxygen is attached to the iron, but the iron must be in the +2 oxidation state, which is known as the ferrous form of iron, Ferrous. If the iron is oxidized to the ferric form, say due to free radicals, then it no longer can carry oxygen. Carbon dioxide, on the other hand, is carried on some of the basic amino acids in globin. And we will discuss this more in the respiratory section. Okay. The production of red blood cells starts, as we did before, as from the hemocytoblast. The hemocytoblast then differentiates into something called the proerythroblast. The proerythroblast then differentiates into something called the early erythroblast, intermediate erythroblast, late erythroblast, reticulocyte, and finally the mature red blood cell. There are other classification systems for the development of the red blood cell, but I'm using one that's more simple. As you can see, when you go through my PowerPoint slides, here's erythropoiesis, which means the development of the red blood cell. Red blood cells are produced on average about 2 million per second. What does that mean? That means that you're losing 2 million red blood cells per second due to destruction. As I alluded to, we have the proerythroblast, early, intermediate, and all through the process, various changes are occurring that you can look at in my PowerPoint. The reticulocyte is the last stage before the mature red blood cell. At that time, the nucleus has been taken out, has been extruded. Thus, the center caves in, giving more surface area. You still have some ribosomes, and you still have some mitochondria. The red blood cell is generally released from the bone marrow in the mature form, but a few are released as reticulocytes, and that's what we call the reticulocyte count. Remember, for red blood cells, there's about 4.8 to 5.2 million per microliter. So some of those are reticulocytes. Generally about a 0.5 to, let's say, to 2% may be reticulocytes in the circulation. Here again is a picture of this. The regulation of erythropoiesis occurs using several paracrine secretions and hormones. But the most prevalent hormone involved in red blood cell production is something called erythropoietin. Erythropoietin is the main hormone. Erythropoietin is primarily produced by the kidney, but also some produced by the liver. How does the kidney know to do it? The kidney is measuring your blood oxygen level. If the blood oxygen level goes down, then the erythropoietin level is increased to attempt to produce more red blood cells so that you can carry more oxygen. If the oxygen level in the blood gets higher, then the erythropoietin level goes down. So the kidney is indelibly tied to the production of red blood cells. The liver also produces some erythropoietin, but the kidney is the primary producer of that. And again, as I alluded to, it's measuring the blood oxygen level. So again, here's erythropoietin. Hypoxia, which means low oxygen of blood due to decreased red blood cells, then all of a sudden the kidney would kick up its production. Okay. The requirements for red blood cell production are proteins, lipids, carbon, the same thing you would have for cells. But when you look at what the red blood cell needs a little bit more than any other cells is iron, vitamin B12, and folic acid. Thus, those particular substances, when they decrease in amount in the body, can cause a low blood count and potentially anemia. Okay. Red blood cells live about 120 days, we should say, plus or minus 20. Their primary destruction is due to them circulating through the bloodstream and eventually getting old and losing the flexibility to be able to transverse through the blood vessels. The spleen is the primary organ that plucks red blood cells out. In the spleen, you have splenic macrophages that heavily look at the red blood cells as they go through the spleen. The course through the spleen is very tortuous. Thus, red blood cells must be able to significantly bend through the spleen in order for them to keep living. When the splenic macrophages see that red blood cells are having difficulty transversing through the spleen, those macrophages will pluck the red blood cells out using phagocytosis. When red blood cells are plucked out, they engulfed by phagocytosis. Lysosomes within the macrophage will come in and break the red blood cells down. Essentially, it will separate the rest of the cell from the hemoglobin. So the hemoglobin will come squirting out almost like ink from a squid. And when it does, then the hemoglobin inside the macrophage will be broken down into heme and globin. Heme and globin is two components. The globin, which are proteins, will be broken down to their basic amino acids, and some of the amino acids will be used by the splenic cells, and others will be transported to be used in other areas of the body. If you have no need for these particular amino acids, then amino acids are destroyed in the liver primarily and then broken down to something called urea. Okay. Now we have the tetrapyral rings. The tetrapyral rings, which is the heme. From the tetrapyral rings, the heme is broken down. The iron is plucked out. The iron is plucked out, as you can see, fate of iron. And then, as you can read, the iron is transported around to cells that need it. And is stored or, in some cases, utilized. The tetrapyral rings are broken down through several steps, ultimately forming something called bilirubin, which is called yellow bile pigment. The bilirubin then is placed in something called bile. Bile is made by the liver and stored in the gallbladder. This bilirubin is placed in bile, as you can see, as one of the components. However, bilirubin does no function of bile. Bile's job, that we'll talk about at another time, is to emulsify fats. It allows the intestine to dissolve fats in the water layers for absorption. So the breakdown of red blood cells involves bilirubin. So that would then also encompass a discussion of jaundice. But since I'm doing an online course, I will not discuss jaundice in this particular course. All right. So we pass by all of these, and then we get to blood transfusions. In blood transfusions, that's when we decide if we take whole blood, and whole blood can be separated into plasma. In the clinical facility, plasma can be stored as something called fresh frozen plasma if a person needs plasma. The rest, which would be the formed elements, you can store the red blood cells. The red blood cells are stored as something called packed red blood cells. The white blood cells are thrown away because one person's white blood cells cannot be used in another person's white blood, another person. And then the platelets can be stored also. I go now to where are the red blood cells produced. Okay. Red blood cells are primarily produced in the bone marrow. You have two types of bone, let's say, flat bones and your long bones. Most production of red blood cells occurs in the flat bone. So if a person has to have a bone marrow aspirate to look at their cells, generally we will go to the flat bones. Now, of course, the flat bones would be the ribs, the sternum, the pelvis, and so forth. Most of the time, a blood marrow aspirate is done from the pelvis because it's a larger bone, unless there's an infection in the skin there. Also, it could be obtained from the skull, but the skull obviously is a dangerous area to push a needle through because it may go all the way through to the brain. So that's where red blood cells primarily are produced. And as I alluded to earlier, they generally are released from the bone marrow in the form of mature red blood cells, but some are released in the form of reticulocytes. I encourage you to read in the text pages 638 to 644 in the Mariap text to get a further clarification of this, and also to look at my PowerPoints as you see. Thank you very much.